want to start with two things. One, I'm kind of bummed that I have to give a talk right now because I like to just sit there and think about Andy's talk. Two, my talk is also a result of a panic spiral. So yeah, I'm glad this is a safe space. Okay. So yeah, I introduced myself earlier, but my name is Loes Olde Loohuis. I'm faculty here. I've done the CGSI drill before. I should know about all the hardware. My primary appointment is in psychiatry, but I also have appointments in computational medicine and human genetics. Today, I'm not going to talk about psychiatry at all. Today, I'm going to talk about inclusion bias and how it affects common variant analysis, genetic analysis in health system linked biobanks. And this is actually a follow-up talk of a talk that I gave last year, which was, this was my title slides. You see, I was a lot more in a state of panic at that time. I was like, can we use biobanks to study people that are not in it? Now I'm a little bit calmer and I've understood a little bit better. But basically what we all do is do a lot of genetic analysis in biobanks, and we take phenotypes and we take people with a certain phenotype with another phenotype, and then we do genetic analysis comparing them. And what I was worried about for a long time was if I take one case group and another case group and I do genetic analysis, does it matter how those people came to be part of my biobank? And are there people that are not in the biobank that could, somehow affect the genetic findings that I see? And so, yeah, we started to really dig into this. And by we, I mean Aditya Pimplaskar, who isn't here. He should really be giving this talk because not only did he do the work, he also made majority of the slides that I'm going to show today. He's actually not in LA, so you'll have to do with me. But he really led this project. Okay, so biobanks are the gateway to precision medicine. We all love them. We all study them. We're here together because we study them. We combine EHR data with genetic data or genomic data to do all kinds of things. GWAS, subtyping, G by E, precision medicine. And we're very excited, and I'm very excited about the promises that these biobanks bring. They're getting bigger, they're getting more international, they're getting more representative of different populations, and that's all very exciting, including we have Atlas here at UCLA, which is one of the world's most diverse biobanks. So that's great, but most biobanks, including the one at UCLA and really the vast majority of biobanks, use opt-in consent. You're not automatically part of the biobank. You may be part of the, registry if you live in Finland or in Denmark, and even there's some opt-out protocols, but UCLA Biobank and the vast majority of EHR-linked biobanks have opt-in consent protocols and various recruitment strategies, like how do you approach patients, asking them if they want to be part of the biobank. And they might not be consistent across biobank settings. They might not be consistent across time. We might not know anything about the biobank's recruitment protocols. Like, for example, all of us, many of us use all of us, but how really all of us came to be is a little bit of a mystery. And so the question that we asked is, do these decisions about these protocols impact downstream analysis? Because obviously what we're worried about is that you can have some genetic feature and some outcome feature both being correlated with enrollment and therefore induce spurious associations that are not really what, part of the mechanism that you're interested in. Okay, so two questions. Can we measure the induced bias without knowing changes in recruitment strategies? If there's bias, does it really matter that much for our genetic analysis, and can we correct for it? So, for example, this is not some kind of new or novel idea. This is like what people in epidemiology think about a lot. Also, people have looked at it in relation to UK Biobank, where they compared UK Biobank participants to census-derived UK individuals, and found that there was all kinds of differences between UK Biobank participants. They were healthier, more likely to be women, etc. They also showed that that affected genetic analysis. But now we have sort of an added benefit is clinical context, because we have the entire EHR. And so we can see the people that are in the biobank versus the people that are in the EHR but not in the biobank. How are they different? And we have a lot of ways we can measure those differences. And so that's exactly what we're going to do. We're using EHR from UCLA for the entirety of these slides. We have clinical records for about 4 million patients, depending a little bit on how you count. And about 100,000 people had consented into the biobank at the time that we did this analysis. 55,000 of those, we had genetic data. And so what do we find? On the right, you can see a whole list of features, and you can see differences between people that are enrolled in Atlas, which is our biobank, and the rest of UCLA Health. And with the rest of UCLA Health, I should be a little bit more precise. Those are individuals that could have been recruited. So they had visits, they had care during the time of recruitment. And so there's a whole list of things, and you can see that across the board, there's all kinds of differences between people that are in Atlas versus not. So for example, I have some more details here. Atlas participants are much more likely to receive primary care at UCLA. They're more likely to have private insurance. They have more healthcare utilization. They have more encounters per year than people that are not part of our biobank. They're more likely to be white, non-Hispanic white. They are more likely to be non-smokers. They have higher socioeconomic status. And these are measures, SVI, Social Vulnerability Index, and BAS, barriers to accessing services, that are zip code based. So they're not individual based. But a lower number here means you have less vulnerability to socioeconomic insecurities. And so the Atlas participants are more to the left. And so we're like, okay, yeah, there's clearly these differences. Let's bring them all together. Can we build predictive models of who is in Atlas versus who is not? Given somebody's EHR profile, can we see if you're part of the biobank or not? And we used all kinds of features for that, demographics, but also clinical features like ICD codes. And what we found was, yes, we can really well distinguish enrolled individuals from non-enrolled individuals. Here we have the probability of enrollment colored by those that are enrolled in Atlas versus those that are not. We get an area under the RC curve of 0.85. So like it's near perfect, the prediction.
What was your strongest feature?
Primary care, receiving primary care at UCLA. We did also do a model without, like only including individuals that receive primary care. That model performs quite a bit worse. That was the strongest feature, but many of these features are redundant, and even if you take a feature out, the rest will sort of pick up the variance.
So how was Atlas sort of recruited, right? Or, they had brochures in primary care?
It changed over time. There were many different strategies. Initially, it was in clinic with an iPad, so they came into clinics with iPads. Later, in 2021, they started sending out messages through the patient portal, where you could just click, like you would get an email if you've been.
Would be primary care?
Not all. No, you can have patient portal if you're not primary care. Yeah, enriched. Yeah, and you can see This is our biggest feature. You can see it very clearly, but there's still a chunk, like a third or so of the participants are not primary care receiving. So you don't have to be primary care to be part of Atlas. We did, so here are the important features, is primary care, encounters, ethno-racial categories, smoking status, some diagnostic codes, age, etc. We also looked, because of the different strategies of recruitment changed over time, we looked over time, like 2020, 2021, 2 and 3. In addition to over time, we looked at people with more encounters, because you might say if you have only one encounter, you couldn't really have been recruited. There was too little care that you got from UCLA. And so we increased the burden to be considered like a control, to be not in Atlas. And it didn't really matter what we did. There was quite a lot of stability in terms of what are important features for being included in the biobank. Primary care, is always been a positive factor, but it stands out because it becomes like more important over time. So, well, now that we have the probabilities of enrollment, we can use those as weights to reweight our analysis and see whether that sort of takes care of some of this inclusion bias. So we use inverse probability weighting for that, where the people that are unlikely to be enrolled are upweighted in our analysis. So first of all, just to check that this reweighting actually takes care of some of the signal, here you can see all those features. In orange, you can see the, in this case on the left, the odds ratio of its association with being included. And in blue, you can see what the remaining odds ratio is after you do a reweighted analysis. And so you can see that most of the signal that we see sort of disappears or gets flattened out a lot after we do the reweighting. And on the right side, you have a sort of variance explained. So the reweighting works. So now we wanted to see, like, okay, can we apply this to a genetic analysis where we compare a scheme where we're not weighting to a scheme where we're weighting and see how much it matters. And last year, I was here in the same place, or actually another place that was a little bit cooler. And I was saying, like, how do you know, like, what are true positives? And Lisa said, oh, we figured that out for you. You don't need to make this map yourself, because we made a map, which we call the phenotype-genotype reference map, that has sort of a curated set of known associations between SNPs and Phi codes that you can use to evaluate your biobank. And I was like, perfect. So we used that catalog and just basically did a very simple check where we took an unweighted analysis and a weighted analysis and see how many of those known variants do we replicate. And it turns out that we are able to replicate more variants if we do the weighted analysis. It's a little bit surprising to me because with the weighting, you lose a lot of effective sample size. So in fact, we have less power. I mean, we have smaller sample size, but more power.
Why you use sample size, effective sample size, when you do the weighting?
Because you reweight on both sides, and so the effective sample size is a function of the distribution of the weights. I didn't include it in the slides, but it's quite a big drop. So, but we find quite a lot more replicated associations in the weighted analysis versus the unweighted analysis. And yeah.
I think it's possible that the original discovery cohort is just close unsorted or weighted sample in some way? I'm trying. It's an awesome result.
It's possible, but I think it's unlikely because, because of the representation of individuals in UCLA. But I haven't checked it. It's an awesome result. I was a little bit surprised by it. But if anybody has any ideas for why that is. We also see that, in the weighted analysis, the overlapping ones, they're more significant in the weighted analysis than in the unweighted analysis.
So did you plot like effect size or Z-scores for weighted versus unweighted? You choose a threshold cutoff.
Yeah. We did not compare effect sizes directly because of the weighting we use, because most of these are logistic models, and because of the weighting scheme, we use a linear model, so it's hard to compare the effect sizes directly. We did do a whole chunk of
The Z-scores.
Yeah.
Z-scores, like a scatter.
For this, we could, I don't think we actually have that, but we could totally show that.
This is a variety of different phenotypes as well.
Yes.
It's like just a smorgasbord, and now you have like
all kinds of phenotypes.
Okay,
all kinds of phenotypes.
Was there a certain phenotype that was enriched for the weighted versus not, like maybe continuous, maybe something that's a little more objective or subjective?
No. I'm saying no very clearly. I don't have a slide on this, and I don't remember it offhand, but there was no clear pattern in terms of, we have the slide, like there's a C, which actually shows it per phenotype, but I didn't include it here. There was no pattern in terms of type of phenotypes that were more likely to be replicated. We did look at a bunch of different p-value thresholds, and does it matter, like how significant it needs to be, and we didn't, it's a very stable and robust result. We also, this, I didn't mention this, but this is using a model that only uses features that are not ICD codes or not phi codes, so that we're not like biasing our, not sort of double dipping and including our phi codes in the model and then reweighting by it and then doing an association analysis. This uses only 5 features and gets an area under the ROC curve of basically the same as the full model. So then we did PHIWAs with polygenic scores, and we just picked 5 polygenic scores because they had various sort of genetic architectures: MDD, type 1 diabetes, BMI, rheumatoid arthritis, and happiness with one's own health. So the question was, does it matter for what we find with PHIWAs? And it turns out that it does matter for what we find with PHIWAs. So for each of these traits, you have 3 bars. The ones that are significant for both, like the PHIWAs associations found in both, only found in unweighted, and only found in weighted. So there's varying pattern. So for all of these traits, we see differences, right? There is no trait where everything is blue. No, doing the weighting or reweighting really matters for all of them. But for some traits, the effect seems more striking than for others. For example, here's MDD. Here you see also the actual PHIWAs, both the unweighted and the weighted one, mapped on top of each other as like a Manhattan seascape plot or something. But what we find is that in all cases, the actual on-target Phe code, like in this case MDD or depression or mood disorders, stay strongly associated with the PGS, which is what you expect. You're not going to change it much, but some other traits really vary quite substantially. There's many traits that are significant only in unweighted, so they sort of disappear if you do. The way we think about it is that they disappear if you do this stress test where you like try to see if you reweight the sample, is it still significant? Many of them disappear, but also new ones pop up. This is MDD. This is happiness with one's own health, which is the most striking ones where a lot of associations, 65, disappear if you correct for the ascertainment bias in the biobank. So that was pretty striking to us. And I already mentioned that, the sort of ones that are corroborated are often in the same chapter as, as the polygenic risk score was built for, which makes sense because you have most power there. Okay, so there is bias in the biobank, and it's really strong. Like, you can really tell by looking at somebody's EHR if they are in the biobank or not, which is pretty striking. There's big clinical differences in demographic characteristics that can stratify if you're part of biobank participants or not. Having electronic health records on everyone here is a real plus because you can do very fine-grained analysis. But we're not maybe only interested in comparing the people in the biobank to the people that are in our EHR. We kind of want to see how does it compare to people in LA or people in US. Which, of course, by that, if you want to do that, you have to rely on census or other, publicly available data types, which are much more sparse. We can, take the weights that we derive from these predicted probabilities and adjust our genetic analysis or downstream tasks. We find that replication of GWAS variance is improved under this weighting, and PheWAS results are affected by doing this reweighting. And so the way we were talking about this when we're doing this work is to really think of this as a filter for sort of false positive association, not necessarily a way of identifying novel associations. Of course, this, as a final thing, this is just very vanilla, like the most vanilla thing of inverse probability weighting. It's not going to work for all your genetic analysis. Like if you have a rare phenotype or a rare variance, it's going to be, the skew that you get from the weight, from the distribution of the weights is going to mess up your association. So you need common traits and common genetic variance for this super vanilla thing to work. That's not to say that you cannot come up with something smarter that will work for rare traits and rare variants. So I think that's just an encouragement for you guys to work on that and figure out how to do that. One other thing that I should mention is we are considering only inclusion bias, like who was included in Atlas, who was not. We have not separated recruitment bias from participation bias. Like who gets asked, do you want to be part of the biobank versus who says, yes, I want to be part of the biobank. And these are two different things at play. And one of our next steps is to actually separate them. We've applied for this data because there is, knowledge of who declined to consent to be part of Atlas and who was offered. We just don't have that yet as part of our EHR. But I think that will be very important because knowing what the different biases are in these different steps can inform recruitment strategies and especially, allow us to develop strategies to include more people that are underrepresented in the biobank. with that, I want to thank my team. It's mostly Aditya who did a lot of the work together with Joanne. Bogdan is a collaborator that was part of this. We miss him dearly, but he's still, with us in spirit. And Veronica and Jeff. The paper is online, but we have quite an updated version based on reviewers' comments. And so if you are interested, just email me and we'll send it along. Questions?